Welcome to another episode of Behind the Research.
From my heart and from my hand, why don't people understand my intention? Weird science, elastic tubes and pots and pans, bits and pieces and the magic from my hand will make it weird science. Things I've never seen.
Our guest today is known to the dark halls of San Francisco State. Her vivid personality and bright humor bring light to foggy San Francisco. I am talking about Talia Hart. Thank you for coming to the show. Introduce yourself to the viewers.
I'm from Los Angeles, California, and I moved up to the Bay Area to attend San Francisco State University. And I currently study under Dr. Carmen Domingo.
What are you studying in the Domingo lab?
So I studied development of Xenopus laevis embryos, and I examined the plasticity of neural and muscle cells.
Oh, fascinating. Can you elaborate on that?
Sure. So my project aims to better understand the role of cell plasticity on cell fate in early vertebrate embryos. So specifically, I look at temporal and regional factors that influence the patterning of neural ectoderm. So I use a cell transplant approach, which is when you take tissue from one embryo and you graft it into another embryo. And I use this technique to examine how neural cells can change and adopt into a muscle fate through grafting in specific regions and also at different stages.
Oh, I see. What are some obstacles have you faced in the Domingo lab?
It's a good question. Transplants, transplants, transplants. I remember last summer when I was given full rein on my transplant experiments. I was extremely nervous because it requires a very steady hand, and I had just finished about 4 cups of coffee. I must have mangled at least 20 embryos just preparing them for my experiment. But with the guidance from my mentor, Chrissy Tejas, I was able to obtain the patience and steady-handedness that's necessary for this project. But I have to say the most challenging obstacle would be to isolate the neural ectoderm tissue. I have to carefully extract it as to not contaminate it with epithelium tissue or mesoderm tissue, but that just gets easier with time.
What did you learn from that?
Basically, I learned each failed experiment is an opportunity to learn and a challenge to do something that I've never done before. It has given me the confidence and aspiration to accomplish more as a scientist.
So you have been recently chosen to become a fellow in the Choose Development program. What do you plan to gain from this?
I hope to gain more of an insight in the field. I'm also excited to learn about other people's projects that may or may not pertain to my own research, giving me a more comprehensive understanding of development. I also hope to hone my research skills and techniques to prepare me for graduate school and overall contribute to meaningful discoveries in science.
Wow, that was beautiful. Well, it looks like that's all the time we have for today. Thank you very much for stopping by.
Thanks, Chris.
Tune in next week where we'll interview Buddy the dog, who has recently adopted 2 frogs after they lost their mother to research. Stay classy, SDB.